of Pharmacy, Kolhapur. He has completed his M.Pharm in Pharmaceutical Chemistry and PhD under Shivaji University, Kolhapur. His industrial research projects involves managing drug discovery pipeline, which includes structure-based drug design, QSAR, and molecular modeling, analyzing and deciding protocols for preclinical and clinical testing, executing of protocols and analysis, preparing pre-formulations, filing patents, and interfacing and technical interactions for the flagships products of Relife MDS. He has international publications of about 25, and he has been an invited speaker for various national and international conferences and workshops. So we have with us a modern researcher and a computational chemist for this FDP who shall give some insights on predicting drug pharmacokinetics applications in modern day drug discovery. Over to you, sir.
Yeah. Hi, this is Kundan here. Am I audible?
Yes, sir. Your voice is audible, sir.
Okay, yeah. Thank you for the confirmation. Yeah. So can you make me the presenter so I can share the screen?
Yes, you are presenter, sir.
Okay. So first of all, I would like to thank the organizers, Dr. Manish Bhatia sir, the entire staff, principal Dr. Rachin Murray sir, and all the people who are working for making this workshop go ahead. So I just would like to ask, is my presentation visible?
No, sir, your presentation is not visible. Share your screen, sir. Yes.
Is it visible now?
Yeah, it's visible.
Okay, yeah. Thank you very much. Okay, so so first of all, good afternoon to all the participants and the organizers. And I'll start with my presentation on predicting drug pharmacokinetics and its application in modern day drug discovery. So my usual presentation would start with this slide itself. What is the problem in pharmaceutical R&D? I hope Dr. Murumkar sir has already given a lot of details about the industry and how the drug discovery process goes on. So I would just like to add on to it that so when we talk about traditional drug discovery, we require around the pharmaceutical industry requires minimum 10 years of time for the drug development, and it would cost up to around 2.5 billion US dollars for the total development. And statistics have suggested that almost 75% of this cost of the R&D is attributed towards failures. So you can understand that if we are investing around $1, 75 cents are going into failure. So only 25% or 25 cents come into as a success of the pharmaceutical industry. So it has been various people from the industry, from the academia, all over the globe have been suggesting various different solutions, including the AIs, including the technologies, and many other tools for overall the pharmaceutical industry.
Ingale, sir, I'm sorry to disturb you, sir, in between. But there is some disturbance in the audio. Can you please check out at your end?
Okay, just give me a minute.
Yeah.
Is it okay now?
I think bit okay.
Okay. Okay. Let me just change my audio device.
Yes. Yeah. Okay.
There is a heavy echo.
Okay. Okay, so just a minute. I'll change my audio device so that it's better.
Yeah. Okay, sir. No problem.
Hello.
Yes, sir.
Is it better now?
Uh, hello.
Hello.
Yes, sir. Can you speak just one or two
Hello.
sentences? I'll just check it with.
Yeah. So is it better now? The quality of audio?
Yes, yes, sir. Better than before. Much better than before.
Yeah. Okay. Thank you and sorry for the disturbance.
No, it's fine, sir. Thank you so much.
Yeah. So we were at the stage talking about that various researchers across the globe have given different solutions so that the total R&D development cost or the amount of the R&D cost that goes into the failures is reduced. Now, AI, artificial intelligence, various computational tools, including molecular modeling. Then you can also have pharmacokinetic tools. You can also have different clinical simulations which can be done. So overall, these simulations, all the computational simulations that are being proposed, I think different people will be talking about these different innovations and different tools in this workshop as well. So these tools, all these tools are, people are looking forward that these tools would contribute to increasing the percentage of success, or if not increasing the percentage of success, at least reducing the cost that goes into the failures. That would be a great turnaround for the pharmaceutical industry. Now, if we look at what a bit details about what are the problems. So if you see in the first image, I have given attrition rate, that is number of compounds that are lost in different stages for one molecule to come into the market. So if you look at the first bar, that is the discovery bar, around 1500 compounds are lost in the discovery phase, approximately 9 compounds in phase 1, 5.5 in phase 2, 1.8 in phase 3, and 1.3 in registration, and finally one compound comes into the launch stage. So you can see, ideally, when you start with around, let's say, around 2000 compounds or even more in the practical discovery stage, in virtual discovery, the number would be even very high. So you can start with around 10,000, 10 million compounds, and finally you would reach to one compound which is launched into the clinical stages. Now, people have also found out the reasons why, what are the reasons for these failures. So you can see a major chunk is ADME PK. So it contributes to around 39% of the total failures, while efficacy turns around into 30% failures. Now, if we try to uh, uh, evaluate both of these efficacy and ADME parameters, you can see that ADME in turns is resulting into the efficacy. So if your drug molecule is not if the ADME properties are not according to its requirements, then it will have a substandard efficacy, and finally it would lead to a failure. So total, let's say, you can say around 69, almost 70% reasons of failure could be due to ADME and PK and efficacy of the molecule. Now, considering these statistics, what would be the probable solutions? Okay, so before going to solutions, let me just take over take over a brief drug discovery workflow. So in initial stages, so you can see the drug discovery stage would be classified into different stages: the initial stages, the mid stage, and the final late stages. Now, in the initial stages, we have to perform different activities which are protein related, chemistry related, prediction related, and interaction related. So protein related would be confined to target modeling, target validation, active site identification, and structural comparison. So I hope different speaker would be talking on that. Then there would be chemistry related, which are more related to the drug molecule structure, where you can see where the lead designing, lead optimizing, what I'm talking about could also be a repurposed drug, uh, which is the current hot topic, and I'll be talking about it also in subsequent slides. Then there are prediction related. I overheard our previous speaker, Dr. Muramkar sir. He was talking about prediction. It could be AI-based prediction. It could also be molecular modeling based prediction in terms of activity. He was also talking about QSAR, which could be solubility based predictions or ADME tox predictions, then CYP binding and hERG predictions. There could be interactions related to protein ligand, protein peptide, and virtual screening assays. So here in this presentation, I would be focusing more on ADME tox prediction. So when we talk about pharmacokinetics, it's mostly we deal about the ADME properties. Now, let's go and see what are pharmacokinetics. I know all of you attending this seminar would be faculties in different colleges, and they have a good understanding about pharmacokinetics and pharmacodynamics. Just so that this slide is just an introductory slide so that we can dive in deeper. So we'll see how all these are related. So pharmacokinetics, as we all know, how the drug travels inside the body, while pharmacodynamics is what drug does to the body. So it could be in terms of cell death or biochemical changes, differentiation, proliferation, or anything. Now, when you combine both of these, pharmacokinetics and pharmacodynamics, you can also estimate what are the toxicities of your molecule. Now, when we are going to talk about pharmacokinetics, usually, so as a layman, so let's say I would, so don't mind please if I take some small examples in between. So as a layman, and let's say some layman is suffering from a headache and he takes an oral tablet. He would be I don't know how many of them wonder that okay, I'm taking a tablet which goes into my stomach, and how does it finally uh, relieve my headache? Okay, so similarly you have different routes of administration. So you can see a small hippo over here. The mice over here are pretty larger than the hippo, but they are wondering that if how would this
Hello,
hello
Kundan sir.
yeah
Sorry to disturb you. That sharing stop sharing option is there. Hide option is there.
yeah okay
Okay, hide it.
okay so the mice are really wondering how could that injection given to him is going to cure his sore throat. Okay, so basically it is the same question we are trying to address, that whatever drug we are going to administer, how it is going to go and relieve the problem that we are going to or problem that the drug is supposed to resolve. Okay, so just a brief snapshot of what goes in and how it goes. So you can see, as we all know, that when you take a tablet or a capsule, it goes into the gastrointestinal tract, and then finally it is absorbed into the systemic circulation, from where it goes into different tissues. It gets metabolized into different organs like liver and kidneys, and finally through the kidneys or maybe other routes of excretion, it will get excreted from the body. Now, at across these different stages where the drug travels from its site of administration to the site of action, there are different processes which go on. Now, the let's say if you start with the initial stage of administration, what are the properties that would be important for a drug molecule or when you are deciding what should be the route of administration or how the drug should be administered. So there you can consider what are the different drug related properties, what route is feasible. So let's say, for example, if a patient is in if a drug molecule is to be administered to a patient who is in ICU on a ventilator, then he cannot take an oral tablet. So what should be the route of administration, what dosage form can be easily administered to the patient, when the drug is administered or when the dosage form is administered, how it will enter the systemic circulation. If it is not IV, it has to cross different barriers like gut. If it is a topical, it has to pass through the skin, and if it is a drug which has to act in the lungs, it has to cross all those barriers to reach into the systemic circulation or the site of action. Now, while the drug is passing from the site of administration into the systemic circulation, we also have to consider one more factor, which is the stability of the drug molecule at the site of absorption or at the site of administration. So if a drug molecule, when you're administering oral, and if it gets degraded, how it would go into the systemic circulation. So these are different problems, along with once the drug molecule enters your systemic circulation, then it also has to transport it. It also has to be transported into different tissues. So these tissues, to enter that, it may have to go to a passive diffusion or an active transport. If it has to work into the brain, it may also have to penetrate the blood-brain barrier. So in all, when we talk about a drug reaching from the site of administration to the site of action and finally elimination, there are various microprocesses that go into the, or that are happening into inside the body between the drug as well as the biological systems of the body. Now, finally, when the drug reaches at the site of action, it should not happen that the drug reaches, but the drug concentration at the site of action is suboptimal. So overall, all these ADME properties and the route of the drug administration and the drug related properties, they govern the concentration of drug at the site. Now, when we look at a typical pharmacokinetic graph, sorry, just a minute, I'll try to add a pointer over here so that okay, fine. I'm not sure how it can be added. Yeah. So if you can see the mouse cursor over here, you can see this is a total typical pharmacokinetic plot of a molecule. We'll be discussing it. So here, from the side, from the time drug is administered, it takes some time to reach into systemic circulation, and then for certain time, the concentration increases. It reaches its maximum level, which we call as Cmax, and then starts decreasing. And finally, so this decreasing, when it starts decreasing, it enters into the elimination phase. Now here there is a small orange line, dotted line. So this is the effective concentration that is required for the drug to be active. Now, what are the things that are important over here? First is the time for which the drug concentration remains above the effective concentration. So let's say if your drug concentration remains for 1 hour or 2 hours above the above the effective concentration, then you need to administer multiple doses so that it continuously remains above the effective concentration. What is the Cmax to EC50 ratio? So this Cmax to EC50 ratio or effective concentration ratio is also important. Higher the ratio, you can assume that for more time, the drug will remain above the effective concentration. The AUC to effective concentration ratio also in terms dictates for how much time the drug will remain above the effective concentration. And if your molecule or drug does not remain above the effective concentration for a sufficient duration of a time, then its overall clinical efficacy would be very limited, or there would be different problems in terms of frequency of dosing, or it could also be in terms of increasing the dose of the molecule, which may not be feasible. Yeah. Okay. So I hope you can see the screen as well as you can hear the audio, right?
Yes, sir. Yes.
Okay. Yeah. Thank you. Now, this is a preliminary for our understanding that what are the pharmacokinetic applications. So, so as we saw, what is the bioavailability? You can measure the bioavailability. You can also plot a dose-response correlation. So, as the dose increases, is the is there any increase in the response? What is the effect of physiological and pathological conditions? So, we all know that what are the different parameters that affect the ADME properties. So if a patient is a disease patient, then the PK profile would be altered. Then the pharmacokinetics could be used for dose adjustment. So as I said, in case your drug concentration is not above the effective concentration, then you may have to adjust the dose. Then evaluation of drug interactions and toxicities. So we'll see how this can be done subsequently. And it can also be used for clinical prediction. So clinical prediction could be in terms of the dose of administration, the entire dosing regimen, or what could be the effect of co-administrations. So there would be multiple drugs which are going to be co-administered along with the drug of interest. So what could be the effect of that? So all these could be done through pharmacokinetics. Now, a typical workflow for pharmacokinetic predictions would be like, initially you need to have an in vitro or in vivo assay data. So that assay could be in terms of permeability, solubility, or in vivo PK in some animals. Then you could have in vitro hepatocyte or in vivo PK data. Again, sorry, it got repeated. Then you could also have some physicochemical properties calculated for the molecule. So any prediction, as is the case, is dependent on the initial data. So some experimental data is always required along with the some experimental data is always required to start with the prediction. Once you have a certain experimental data, then you can start with the predictions where you can predict the bioavailabilities. So there could be more parameters as well. I have just listed very few. It could be bioavailability, which is F, clearance, then the volume of distribution. Using these parameters, drug parameters, you can go for an integrative modeling kind of approaches where you can do a PBPK modeling, which we'll be discussing in details subsequently. Once you have the a kind of integrative modeling, you can go ahead and make an entire PK profile for a drug molecule. Now, this PK profile would be very helpful in various applications. So it could be in terms of label information for the drug, that is how the drug should be administered, what should be given, what should not be given, or it could also be in terms of it could also help in terms of dosing regimen and toxicity estimation. So this is a typical traditional workflow which would be followed for estimating the PK profile. Now, if we come to physicochemical properties, I have listed a very short number of physicochemical properties over here. So most important physicochemical properties that are utilized in pharmacokinetic modeling would be solubility, which is log S. So I'm not going to go into finer details of these properties because I assume you all would be knowing these properties. I've also listed some tools. Some of these would be freeware, some of these would be licensed softwares. So you can also go maybe take a screenshot and maybe use later for your research purposes. So solubility is one important parameter which governs the drug pharmacokinetics in terms of its absorption and distribution throughout the biological system. Then partition coefficient, which also has a huge impact on transport of the drug molecule. This is then the distribution coefficient, that is log D, which is also utilized in the transportation, especially where pH is a factor. PK, which also which is an influencer of lipophilicity and solubility, and protein binding and drug permeability. Okay, so once you have these physicochemical properties predicted, using these physicochemical properties, you can further, using the softwares which I have given earlier, you can do prediction of the different aspects of ADME. So you can predictions of absorption, you can make the predictions of distribution, which could be predicted or which could be used for predictions of these particular parameters. Now, once you have these predictions of ADME, what you would be requiring for these predictions? Okay, so I have listed some of the prediction methods. So traditionally, we use compartmental modeling and non-compartmental modeling. So these are traditional methods which we have been using and studying for as long as I can remember. While I was in studying my pharmacy course, I have also studied this compartmental and non-compartmental modeling. So these compartmental modelings, as you know, these treat biological systems into a different way, where the compartment model divides the body into different compartments. Well, non-compartmental modeling assumes that the biological system is a homogeneous system and goes on. I'll not be talking about these two systems. I would be talking about the next two systems, that is population pharmacokinetics and PBPK models. So population pharmacokinetics, it's very interesting approach for pharmacokinetic prediction, where you can using a limited data, or you can say extensive data, you can map the pharmacokinetics to a huge population. So this population pharmacokinetics is very useful when it comes to predicting clinical response of a drug molecule in a wider populations. So these populations could be restricted to a local area, let's say Indian population, or a Japanese population, Chinese population, white population, and so on. It can also be used across all these different populations. Then PBPK modeling is physiology-based modeling, where which involves a lot of compartmental modeling also, where it considers that your body or the biological system is a set of tissue compartments which are interconnected. So wherein the blood will flow into these different compartments, carry the drug, and there would be different processes occurring into in that. Okay, so we'll go and see in details these two methods. So first method that I would be talking about is population models. So I'm taking population model before PBPK because population models also have a key role to play into the PBPK model. Yeah, so so when we are talking about a population model, as the name itself suggests, that these population models can map the pharmacokinetic properties, especially absorption, distribution, to a characteristic population. So this, as I said earlier, these populations could be in terms of ethnicity, it could be in terms of age, it could also be in terms of any specific disease area. Okay, now why do we need these population kind of methods, or population pharmacokinetic methods? So when we are estimating pharmacokinetic parameters, there is always a inter-individual variability as well as intra-individual variability. So let's say so just to make it clear, let's say for example, one person is taking a drug, let's say for example, as we are into the pandemic era, and we are doing an online workshop just because of COVID-19. So so let's say I would try to incorporate more examples which are more similar to the or more relevant to the COVID situation. So let's say a COVID patient is taking one drug, let's say for example, favipiravir, which is orally taken, and he is taking the drug for let's say around 5 days or 10 days regularly every day, whatever dose has been prescribed. So is it okay to assume that the drug, as the person takes tablet, the same Cmax will be reached every day? It may not be possible every day, depending on what is his diet, what is his water consumption. The the pharmacokinetic profile will change daily. Also, it cannot be assumed that different individuals, so let's say I am taking a tablet and somebody else is taking a tablet, we may have different concentrations or different PK profile. So this in total contributes to intra-individual and inter-individual variability. Also, when there are measurements, so if you are doing a practical peak pharmacokinetic measurement, you can also see that the method that is being used for measurement may also have some errors. So if the error is large, or if there is as we know, it could be a manual error, it could be an instrument error, it could be an environmental error, or the sample got contaminated, and there could be anything that could be happening. So all these problems could be resolved with the help of population models. Now, what are the advantages of these kind of models? So these population models can, the most important advantage here is it could, even if you have a very sparse data or intensive data, it can be sampled into a, or it can be simulated into a population model. This is very important because it reduces the cost. As I said earlier, that if the industry would be looking at, even if not increasing the success rate, but also if we are able to reduce the cost that goes into the failures, would be very, very important, very, very useful. So if you have a very limited data, a limited this kind of models could also be practiced with a limited data. So if you have the company may have to do a very limited sampling, and that would also can be used to map it to a population. Then, as I said earlier, it could be mapped to different kind of special populations, let's say like neonates, in terms of pediatrics, elderlies, or AIDS patients, or critical care patients. So you can build a model for the healthy subjects or healthy populations, and also can further transcribe it to these special populations without any ethical or medical concerns. This is very important because the regulators are also accepting the computational models. I'll be talking about what are the criteria for acceptance later. Now, as I already said, it should be, these kind of models are very cost effective. Rather than doing a PK simulation experiment on, let's say, 100 subjects, I can do it on 5 subjects or 10 subjects and map it to a population model, and then get the data or generate a data for a huge population. So it really saves a huge cost for the pharmaceutical industry. Now, applications. So most of the applications we have already discussed. You can get a quantitative assessment of pharmacokinetic over huge population. These population models could also, unlike any other PK modeling, can help you in defining the dosing schedule. You can also communicate these results to the regulatory bodies. They are acceptable. Then understanding the dosing regimen that I have already talked. You can also have, one of the important thing is reasoning for difference between expected and the outcome in terms of inter-individual and residual. So I heard somebody asking a question that are the in silico results mapping to the in vivo results? So yeah, they do map. There are some differences in those results. I prefer it calling residuals in the computational predictions and the actual results. So there are definitely some residuals, but they are definitely within a certain acceptable range. Okay, so now we'll see what are the different methods which are applied in the population models. So these are fairly popular methods. Naive average data approach. So this is a very simple approach where the analysis is performed by computing the average value for each sampling time point. So what is done here is basically there are, let's say, 10 subjects in the PK population. All the subjects will have a same sampling time point which is predefined, and using the same sampling schedule, the data is collected and that data will be fit to a bigger population. Now here one one assumption is that all the individuals would have which are being utilized as a population in this study would contribute to or would cover the entire diversity. This is the assumption over here. So this method may have certain limitations. Then the next method is naive pool data analysis, where the data from all the individuals is considered or is assumed to be coming from a single individual, and then it is being mapped to the population. So again, here pooling itself may have certain disadvantages, and that's where this method may also have certain limitations. Then there is one more method, which is two-staged approach, where individual parameters are estimated separately. So in the above two methods, we are clubbing the individual data. In the two-stage approach, the individual data is treated as an individual data. It is fitted as an individual data, and finally, uh, the the data that has been obtained, the pharmacokinetic data that has been obtained at the end of the sampling is averaged, and then variances are calculated so that we can establish a range within which this data would vary. Then there is a mixed modeling effect approach, where the approaches that are followed in the two-stage approach, they are simultaneously done. That is, the data is collected and simultaneously it is fit to the population. So this method is fairly popular in estimating the population pharmacokinetics. Now here certain parameters are calculated. I have added these parameters because these parameters would be used somewhere down the line in our presentation, where we calculate theta, which is a population estimate for the PK parameter. Then we calculate eta, which is inter and intra individual variability, which is very important, so that we know what is the variation, and finally what in the outcome that or the prediction that we are going to get, how much variability we can expect. Similarly, epsilon, which is also a residual variability. These three different parameters are these are the major parameters. No doubt there are other parameters also which are calculated, but these are important parameters because they will be helping us in terms of defining the variability in the individual and the population responses. Now we come to the next part of the topic, that is PBPK, which is nothing but physiologically based pharmacokinetic model. Now these are models which are widely being used in the discovery process. Now there are some very good advantages of these models. Now so especially in the situation we are right now, the COVID situation, in the pandemic situation, where finding a new drug at a high speed compared to the conventional discovery method is very essential. In this kind of situation, these kind of models are very helpful, where we are basically repurposing the existing drugs for a new indication, and there is already a good amount of pharmacokinetic data available for those drugs. It's not just pharmacokinetic data, you can also have toxicity data available, and then if there is a system which will help you in predicting the response in this special kind of population or a disease, it would be very useful and would definitely accelerate the discovery process to get a new drug, which could be a repurposed drug or a totally a new drug. But a repurposed drug is the focus into for the final approval. Okay, so what is done over here is ideally the drug uptake, that is absorption and disposition, they are quantitatively correlated with the critical biological processes. So what is done is basically different drug related properties which are pre-calculated in terms of physicochemical properties or uh, the physiological properties or the biological properties. These are for the existing drug or population which are already reported into the data are collected, and then a model is fit. I have also added some workflows which we'll be seeing later. Now here, I'm sorry. What we can do is we can incorporate changes into the pharmacokinetic profile which occur due to growth and aging. Now, so let's say, for example, the pharmacokinetic properties of an teenager, an adult, and aged person would be different. So when we are talking about conventional methods, like compartmental or non-compartmental modeling, it is difficult to incorporate all these aspects into those kind of models. That is where one more advantage where pharmacokinetic modeling is, PBPK modeling is very, very important. Now, this modeling, as I said earlier, also it can be used for predicting across different species. So you can relate mice data, rat data, a dog data, or any primate data together, and you can build up a pharmacokinetic model. Now, just as it has various advantages, it also has certain limitations, and the limitations would be, as I said, for the population-based model, limited data is also usable. But for physiological, this PBPK modeling data, good amount of data is required, and that is why I said that this approach is very much useful for repurposing drug molecules for any disease, especially COVID or any other disease that we are interested in. There are a lot of statistics going around, so where understanding the biological processes is also important, along with uh, along with the statistical interpretation. And if the understanding of these biological processes is, and as you all know, we are, as a human, we are evolving, uh, we have been evolving in terms of our knowledge, but yet we are not able to quantify the, or we are not able to completely understand the biological system. So there is certain amount of uncertainty and variability which comes, which computers right now may not be able to estimate, but that still remains a challenge. And maybe down the line, experts like you, when they start coming up into, or when they start using these technologies, will definitely try to resolve these challenges down the line. Now, uh, one more limitation of this PBPK modeling is you need expertise. So the person who is going to develop this model need to be expert in terms of development and implementation of this PBPK models. Now we'll dive into the PBPK modeling, where you can see what are the different building blocks of PBPK model. So firstly, we need anatomy and physiology based data. So wherein anatomy in terms of what are the different organs involved, what are their volumes, what are their surface areas, what is the tissue composition, what are the flow rates. So flow rates are important because the blood flow may not be the same for, let's say, lungs and kidneys and lymph and everything. So these are physiological anatomy and physiological parameters. One more important physiological parameter that is required is expression levels. So expression in terms of enzymatic expression. So what are the CYP expression levels in that particular organ or a tissue is also important. Then physicochemical properties, which are of course the properties of the drug molecule in terms of lipophilicity, what is the molecular weight, what is the PK, PKB. Now, these are individual anatomy, physiological, and physicochemical properties. Along with that, there are some drug biological properties, which properties, which if available, would be very, very important, which would be bioavailability, partition coefficient, which is also listed in the physicochemical properties, but also permeability and what is its Km, Vmax, Kd, these kind of properties, if available, the modeling could be very, very useful. Now, these are the drug related properties. Now, there are some properties which are some parameters which are required in terms of the study protocol and the formulation properties. So the most important is formulation. So what is the kind of formulation that you are going to use? If it's a tablet, what is the what are its dissolution properties? If it what is the administration protocol that you want to predict? So let's say you want to administer the drug twice a daily, thrice a daily, or once a daily. What this is the dosage regimen that would be required, and special events which would be food intake, exercise, and EHC properties. So all these properties or all these parameters would be required to build up a PBPK. That is, sorry, I missed P in the title over here. Yeah, so all these properties, once you have, you can apply the PBPK modeling for predicting drug-drug interactions. You can build up a pharmacokinetic profile for specific populations, such as pediatrics or geriatrics or people with organ impairments or any other thing. Then the most important application is you can make cross species prediction, disease state prediction, food, what is the effect of food, what is the effect of formulation change. So in terms of when we are talking about formulation change, even there are cases where particle size change in the particle size of the drug would be a change in the formulation, because it would change the dissolution and further properties of your drug molecule. Okay, these are the areas where your PBPK would be modeled and applied. Now, so some typical parameters which are required, which we have already seen, I guess. So molecular weight, the log P value, whether your drug molecule is acid, base, or neutral, whether it is what is its pH dependent solubility, what is its plasma protein binding, what is it blood plasma partitioning, apparent permeability, intrinsic clearance, in vitro matrix binding, then the Vmax, Km, reversible inhibition, CYP inhibition. All these parameters are typically required to generate a PBPK model, and that is why we have been saying that it is slightly data extensive model, so that so you require sufficient amount of data points to build up a model. Now, and typically in our kind of industry, people would build a model, PBPK model, in such a flow. So first, you'll require animal data. So if you have the animal data, you can build up a PBPK model with that animal data. Then simulate that data, simulate that animal data, obtain the PBPK profile, scale it up to the humans at the starting therapeutic dose. So in different so if you are applying this data for a new disease condition, you may not be knowing what is the initial dose, so you can play around, so which is not possible in the clinical condition. So so let's say you start a clinical trial with, let's say, 100 mg twice a daily dose, and then you shift it to 200. It's not possible in the clinical stages, but can be done in the computational experiments. And then finally you can decide the protocol in which would be followed in the clinical trial. Now, when we are building a PBPK model, what are the different stages that would be required? So as I said, when you have different kinds of data, that is molecular properties, ADME data, and some software inputs in terms of body weight, or you can say anatomical, physiological data that is required, you can start building the PBPK model. Once the model has been built, you need to verify that model. Okay, this is very important in terms, if you are going for a regulatory submission, or even for applying that data into clinical clinical stage. It is very important that the model gets verified with the existing data, so you can predict something that is already known and match how good is the prediction. Then you go for the model evaluation, so wherein you simulate the model and try to see how it is predicted for a different species or the same species at a different dosing level. Once your PBPK model is done, you can further apply it for drug-drug interaction model building and drug-drug interaction model prediction. So these are this is a typical workflow we'll see for the PBPK modeling. We'll also see how this modeling actually happens. Okay, so here in the first image, just a minute, I'll just take a sip of water. Yeah, so to continue, yeah, this is in the first image that you can see is a typical blood flow that is considered in the PBPK model. So where in the center you can see all the different tissues have been listed, like lung, heart, kidney, muscles, and so on. Then on one side there is an arterial supply, on the other side there is a venous supply. Along with that, now usually when you take up a drug molecule, let's say if you're taking a drug orally, the compartment into which the drug would go is stomach, then small intestine, large intestine, and then it would depending on where the drug gets absorbed, it would go into the arterial supply. If you are injecting a drug IV, the drug would be deposited directly into the venous supply over here, and then there would be a flow. If your drug is being applied topically, the drug starting point would be in the skin. So this is how your administration of the drug can be taken care of, and then finally the flow can be simulated. So arterial, you can see from the arterial supply, the these dark black arrows, they are going into different tissues. So this is how the blood will flow into the tissues. Once it goes into the tissues, it will come outside of the tissues, and through venous supply again it will be recirculated. So typically we try to simulate the blood flow into the biological system. So you can say it's a kind of a multi-compartment model. Along with that, you can see there are small gray arrows over here. So these gray arrows refer to blood diffusion into lymph nodes. So blood, so the drug which goes into the tissues or into the systemic circulation may also seep into lymph nodes. So that is also considered over here. And finally, elimination, that is you can see from the kidney, you can obtain you can input a parameter for renal clearance. Now over here you can see a small what you can say, a small subunit over here, where from organ plasma into the interstitial space. So when from the arterial circulation, the drug enters into the organ, and from there it can go into the interstitial space and further into the lymph node, and the remaining amount would go into the venous circulation. So from this small diagram, I hope I'm able to clarify that drug molecule, once it enters into the body, it just does not flow around. It has to enter, it has to go into different tissues, limbs, interstitial spaces, and at every point there would be some accumulation, there would be some amount of degradation, there would be some amount, certain amount of retention, and all these things considering, we have to estimate what would be the final concentration at that particular tissue or in available into the arterial supply or the venous supply for the drug to show its action. So overall, this entire thing would simulate and give us a detail. Now it is definitely as not simple as this image. We'll see when we see the actual equations how these are calculated, but this will try to give you an estimate of what would be the drug concentration at different tissues or different flows at a particular given time point, or through across the different time points, wherein which will help you in estimating whether your drug concentration is above the required concentration or not. Okay, so once you build a PBPK model, it can be further extrapolated to special populations, as I said earlier. So I have put in two workflows which are more or less similar. First is the extrapolation of the PBPK parameter or PBPK model to the pediatric population. So wherein you can see once you have the data, physicochemical, ADME or anatomical data, which forms the input parameters, they are given as an input to the adult model. So initially the adult model will be built. So based on the adult model, if at all there is any in vivo data, it would be used to evaluate the initial adult model. If your model is able to replicate the adult data, if it is, then go forward. If it is not, you can if you can again if it is not able to evaluate or predict properly the adult data, then you can re-evaluate and optimize the input parameters. If it is okay, you can go and further apply that individual adult model to a population pharmacokinetics. So we have seen the population pharmacokinetic model, so wherein you can combine the individual adult model into a population model. And once you have an adult population model, if at all you have data, you can verify it. If not, you can go and so here you can scale it, and once you have scaled and evaluated, then finally you can evaluate, you can build up a pediatric model. Now when we are going from the adult model to the pediatric model, there would be what would be the changes? A typical question I would if I anticipate the parameters, the physiological parameters that are going to change. So it is in terms of organ volumes, the tissue composition would be changing, the flow rates would be different, and that's how once we have validated it, it could be further transported into the pediatric population. Now once you have modeled, you can go and evaluate it in the, if at all you have any pediatric data available for the prediction. Now similarly, it can also be applied into pregnant model. So as I said, the typical workflow would be similar. You build a non-pregnant PBP adult model. Of course, when we're talking about non-pregnant, it would be an adult model. You validate the model, then scale the model for based on the trimester period in which your drug is going to be administered. Then once it has been scaled to the trimester model, you can simulate and calculate the PBPK parameters. If at all you have data, you can verify the model. If it is if your model verification is successful, it's okay. If it is not, then you can again refine the model. Now what would be the outcome of all these exercise? It would be in terms of you can estimate what would be your dosing frequency, whether at a particular dose the concentration is sufficient or not. Okay, then yeah, so here I'm trying to demonstrate how your PBPK model can be applicable for extrapolation to drug-drug interaction. So in various cases or various disease cases, patients have to take multiple medications. So whether there are any drug-drug interactions could be predicted over here. So there are some drug specific parameters which also include mechanism based inhibition of CYP following a multi-dose regimen. So we need this information, whether your drug inhibits CYP enzyme or is modulated a CYP modulator or not. If it is there, you can use and build up the model. The system parameters, which also like organ volume, tissue composition, and blood flow. Along with that, it also needs abundance of enzymes and transporters. So in any specific disease condition, if the enzyme expression is reduced in any specific organ or is increased in any specific organ, should be if it is known, that would be very much useful in the model. So once you have the data, you can build up the model, you can verify it using the clinical data, and then you can use additional information in terms of verified models for CYP modulators. So let's say, for example, you are giving a drug molecule and it is being co-administered along with ketoconazole, which is a CYP modulator. So what would be effect, or would there be any interaction between this CYP modulator and your drug, could be estimated. Now, these kind of models would give you two outputs. One is the PBPK model prediction in terms of drug-drug interactions, as well as what would be the dosing module. So if at all there are any drug-drug interactions between the two drugs or multiple drugs which are co-administered, what would be ideal dosing regimen that should be followed so that this interaction can be minimized. Okay, so this is how drug-drug interactions could be estimated. Now coming to the equation part, I'm not sure whether I should be discussing these in too much of details, but definitely we'll touch base those models. So you can see we have seen in the PBPK flow model that there is an arterial supply, a venous supply, the flow going into different organs. So typically you would have these kinds of equations wherein if you have a lung interstitial clearance or lung vascular or kidney vascular model, wherein you can see different terms are there, wherein L, there are different parameters over here. So if we look at the lung parameter, this L, capital L, would stand for the lymph diffusion. Then C is for the circulation. So from lung, what would be the amount of drug going into the lymph, and what would be the amount going into the circulation, which would be subtracted by the so this is the total concentration coming in, and the drug amount going outside the lung model. This is again going into different tissues. So likewise, just to sum up these models, so if we are looking at a tissue model, the tissue model will be such that what is the amount of drug coming from the venous or arterial from the arterial supply into the tissue, where it would be distributed, to which whether it would go into lymphatic circulation or whether it would go into blood circulation. If it is a kidney, what would be the role of the filtration happening over there. So all these parameters considered together, and finally what would be the drug concentration leaving the tissue. This would be a typical equation which we would be using in the PBPK modeling. So if you, for the tissues, the equations are fairly small, but if you see the venous or the arterial equations, they are very, very large, because the drug from the arterial supply or from the venous circulation, the drug is going to go into different tissues and would be coming back from different tissues into the circulation. So all those components have been or would have to be minutely modeled into the equation, so that the models can predict the drug concentration into different tissues, plasmas, and wherever you want with a fairly good accuracy. Now, once you have done this exercise of fitting the model, so just to relax a bit, these equations, you don't have to build those equations. There are different softwares which you can use, which already have inbuilt equations. Well, some of them also have facility so that you can build in your own models. Okay, so just to give you some names of the software, I actually should have added those names into the slide. So there are some softwares like GastroPlus or SimSip or Atlas MPBPK or Farm PBPK, which could be used to model PBPK models. Now, once you have done the simulations, what would be the output that you are expecting? So in the first image, you can see a typical time profile, time concentration profile, where on the y-axis is the concentration and on the x-axis is the time. What is the drug concentration? So here the blue line in the middle is the predicted concentration, that is the simulated concentration. These gray lines, one at the top and one at the bottom, these are the mean, plus and minus in terms of standard deviation. And the orange line over here is the clinical experimental data. So you can see there is a fair amount of parity between the predicted model and the clinical model. Of course, you can see it is an ideal case. There would be some cases where the mean, the clinical concentration would be deviating. Now it depends on multiple factors, how you have developed the model, how accurate is the data, and so on. Now, so when I'm talking about this plot, I would also like to mention one point that many times what happens is the physicochemical data is available for the drug molecule, while the concentration that gets measured into the plasma is of the metabolite, which may be the active metabolite or would be the inactive metabolite. Means it's a case to case. So there, uh, what parameters you are giving as an input becomes very critical to, when you get a actual profile. So let's say, for example, if you are giving a permeability of the drug, and um, and on the contrary, what goes into the systemic circulation is the metabolite, then the permeability values of the drug may not be useful. It may be useful up to a certain extent, but beyond that, it may not be useful. When it goes into the tissue, actually the metabolite is going into the tissue. So the permeability values of the metabolite are important rather than the permeability values of the original drug molecule. So this is a kind of PK profile you would get across the time. Along with that, if you simulate it, you can also get what would be the profile at a multi-dosing regimen. So here you can see, I'm not sure if it is quite clear, but if you can see, the plasma mean is in the blue line over here. The plasma mean in terms of standard deviation, gray line is on the top. This red line and green line, these are nothing but the effective concentration. So this green line over here is EC50 and the red line is EC90. So at a multiple dosing regimen, is your drug able to achieve concentration above the EC90 or EC50 is estimated. So here you can see, I have purposefully added this image. So here you can see the plasma concentration that is in the blue peaks is above the red line. So that is the drug or the dosing regimen is able to provide a concentration above EC90 for a sufficient duration of a time. But there is a catch over here. You can see this is specifically the orange line is the lung line. Okay, so the site of action for the drug is in lungs. Now, if you see these orange peaks, they are below the red line. Okay, so that means the drug is able to achieve concentration in the tissue which is above the EC50 of that drug, while which is below the EC90 of the EC90 of the EC90 required for its action. So this kind of PBPK kind of modeling will not only help you in identifying the dosing regimen. Now, usually we take decisions based on the plasma concentration and forget what is the site of action. So these modelings can be helpful in terms of modeling or extrapolating your what you can say, concentrations to the tissues. Sorry, I just want to make sure, is it visible and audible?
Yes, sir. It's visible also and audible also.
Okay, yeah. Okay, so once we know how the PBPK modeling can be used, we have also earlier also discussed that there are certain uncertainties and variabilities. I'll directly when we are talking about uncertainties and variabilities, we should be knowing that, or we should be mindful that computational predictions are as good as the data that is given to it. Okay, so we usually call it as GIGO, GIGO, garbage in, garbage out. If you're giving a data which is substandard, the model will give, or the predictions that will come to you would be equally substandard. So we have to be very careful about the data that we are giving. That now many a times what happens is, so in a typical repurposing kind of case where sufficient amount of data is available, but it may be for a different disease condition, the population would be different, and if you're repurposing it for a different population, there would be certain gaps which may have to be filled in before predicting the model. So that is one limitation where of the PBPK modeling. Along with that, so let's say typical repurposing scenario where the drug which you are using for repurposing was actually approved, let's say around 50-60 years ago, and the data available is based on the techniques available at that time. Okay, so there would be certain errors in those techniques, and if you have a recent data, it would be better to rely on the recent data rather than the old data, because then the differences would be minimal. Okay, and as I also said, that if you are using different populations or different disease populations or different ethnic populations, then there would be differences in the model prediction. Now you may have data when you are applying this model for a repurposed drug, but the data may be limited when you are developing the model for a new drug. Okay, so let's say you have designed a new molecule in your laboratory, you have synthesized, you have done certain amount of activities, and you also have some predicted properties of it. So in such cases, again, as I said, you may have to generate more amount of biological data to use the PBPK modeling. So of course, it is slightly data limited methodology, but once you have the required amount of data, it can be definitely used to build up a model and use it further. Now, so I would just like to give 2 minutes to this slide. So all know that industry has been revolutionizing. So initially, when let's say uh, uh, typically around, let's say the start of 19th century or end of the 18th century, the industrial revolution was just picking up, where uh, where you can say the mechanization was just based on the steam. So you can say basically it was not automated as now, but I would directly jump to the industry 4.0. So right now, with the help of computational tools and various expertise that we have developed, we are able to totally revolutionize the industrial model. In industry 3.0, the computers were significantly used, and they formed a greater part of the industrial revolution. But now we are going a step ahead with technologies such as IoT and machine learning and artificial intelligence. These computational methods can be used means the use of these methodologies can be stepped up, and we can go a step forward. So the pharma regulators have also now changed their stance in terms of they can accept, or they are ready to accept computational outcomes if they are properly validated. Okay, now in this line, if you have noticed, I have shown part of this slide earlier, where where we are looking at the pharmacokinetic evolution. We start with the in vitro data predicting pharmacokinetic parameters, develop an integrative modeling, and calculate the predictive profile. Now, going forward, what could be done is, based on the clinical outcomes that come up, you can further go back and refine your model. This is definitely currently being practiced. And what is the outcome of that is regulators have started believing into computational models. Okay, computational models, if they have a good amount of qualification to their to be backed up as. So if you're talking about a PBPK kind of modeling, then typically a regulator would be expecting two kinds of qualification: the platform qualification and the model qualification or verification. So platform qualification is qualified for the intended use. Okay, this is typically called as to what area you are applying your model. So uh, if it is qualified for that purpose, you can use it. And the model that you have developed or model that you are using, if it is qualified for that purpose also, then you can use it. So when we are talking about platform qualification, it comes in terms of virtual. If you are using a population model, what kind of populations you are using? Is your model able to build up all the individual parameters, like in terms of lung volume, stomach volume, all the tissue volumes, the blood flows? Then when you are creating a individual, whether it's a healthy individual or a patient, then it should be able to mimic the properties or anatomy and physiological properties of that individuals. And it should also be able to populate the data across different ethnicities, in terms of if your patients are Caucasians, Chinese, Japanese, or African, Asian, and all not. Then computational framework. So you need to have a plan for design qualification and installation qualification. So those who are working in pharma may be well versed with these two terms. Then the system parameters that are going to be utilized should also be well understood. Now, when we are talking about model qualification, you need, if you're you need to verify the drug model in terms of sensitivity analysis and parameter identifiability. So sensitivity analysis is in terms of, if you change a minute property of the drug, is it reflecting into the is it reflecting as a change into the pharmacokinetic profile or not? If yes, then your model is sensitive enough. Then you have different approaches to validate or verify your model. So you have bottom up approach, top down approach, or middle out approach. In bottom up approach, we usually start with the physicochemical properties of the drug and build a PBPK model and verify it with the clinical data. In the top down approach, we start with the clinical data and we see whether the clinical data is coming due to the correct physicochemical properties of the drug or not. Middle out approach, wherein we use wherein we start with the physicochemical properties as well as the clinical data or the in vivo data and try to match it over there. Then finally, most important thing a regulator would be looking at is intended use. Okay, what is your intended use and what is its regulatory impact? Whether the regulatory impact is low. So let's say when we are deciding dosing regimen, the regulatory impact is low. When we are trying to estimate the clinical pharmacokinetic parameters, the regulatory impact is medium, and the high impact would be in terms of whether we are trying to obtain, whether the company is trying to apply for a waiver of any BAB studies or bioequivalent studies, or whether it is trying to apply for a waiver for drug-drug interaction kind of studies. So depending on the impact of the use of the model, it should be properly validated. Now, so the question would be that, are there any cases which have been successful with the PBPK kind of exercise? So yes, there are certain cases. So ribociclib, olaparib, these are the drugs for which companies have companies have successfully obtained waivers for drug-drug interaction kind of studies. Then there is a drug called as canagliflozin, which has for which the company had successfully obtained a waiver for an equivalent studies, and so on. So there are multiple instances where PBPK kind of modelings have been regulatorily acceptable. And I hope I was able to give justice and could convey the use of tools for predicting pharmacokinetics and their application into the drug discovery. And with that, I would like to thank. So I would be open for queries.
Thank you, sir. Now I request the participants to please, if you have questions, you can raise your questions. Participants can raise your questions. You can raise your hands or you can put it on WhatsApp.
So usually, just to start with, usually when there are conferences and workshops in person, the lecture just before the lunch is people are pretty desperate for going to lunch. I hope you have not gone for the lunch.
No, they must be there, I think, but must be
Yeah.
feeling hungry. Must be feeling hungry, so might be hurry to go for the lunch.
Yes, yes.
Yes. Anyways, I think there are no questions. It
Okay.
appears. I think. Is there any question by Amol Dixit? Okay, no issues. Uh, Thank you, sir, for giving your valuable time and addressing on this current hot topic. So I request all the participants to rejoin at 3 for the third session.
Okay.
Thank you.
So
Thank you, sir.
thank you, all of you.
Thank you so much, sir. It was a great session, wonderful session, and I hope this will definitely benefit all the participants. And since this video will be available to all the participants on the Atal portal later on also, so
Okay.
whenever needed, they can hear this talk again.
Okay. Yeah.
So thank you so much. Yeah, thank